Welcome to an in-depth look at insulin resistance, focusing on a hidden factor, the lipid signal 2-AG. This is presented by Chip Paul from ECSEducation.Academy, supported by TrueMedX research. The endocannabinoid system, or ECS, is the body's hidden control network for energy, appetite, and metabolism. It uses lipid messengers to regulate these vital functions. What if insulin resistance isn't just about sugar, but about fat signaling inside our cells? Traditional views often lack a unifying molecular cause, but the ECS integrates nutrition, infection, and inflammation. The most powerful lipid messenger in this system is 2-arachidinylglycerol, or 2-AG. Understanding its role is key to unlocking the true cause of insulin resistance. 2-AG acts as a full CB1 agonist, signaling anabolism. It is synthesized on demand, using DAGL from diacylglycerol, making it a critical anabolic signal. Chronic factors like modern omega-6 heavy diets, chronic infections, and inflammation cause our cells to overproduce 2-AG. Pathogenic stimuli, such as bacteria, activate the enzyme DAGL, which drives this overproduction. This overproduction of 2-AG constantly activates the CB1 receptor, sending a persistent store energy signal to the body, even when energy reserves are full. This chronic CB1 activation results in profound metabolic changes, leading to what is called a metabolic gain error. In the liver and fat tissue, chronic CB1 activation triggers lipogenesis, slows mitochondrial oxidation, and most importantly, blocks the insulin receptor's ability to transport glucose. It's as if the body's fuel gate gets jammed, leading to high blood sugar, unwanted fat gain, and eventually the progression toward metabolic disease and diabetes. The effects of 2-AG overload are also detrimental to the pancreas, leading to beta cell exhaustion. Inside pancreatic beta cells, persistent high 2-AG signaling silences the genes responsible for making insulin, temporarily increasing insulin release in the short term, but causing long-term damage. What begins as a cellular adaptation ends in dysfunction, with chronic endocannabinoid signaling causing exhaustion and cell death in the insulin-producing cells. Here is the hopeful part. The ECS is programmable. We can retune it to restore balance and sensitivity. Restoring proper ECS tone is essential for restoring insulin sensitivity. We must focus on the lipid pathways to control the underlying metabolic disease. Balancing omega-3 and omega-6 fats in our diet is crucial, as modern diets create a huge imbalance. Omega-3s help restore proper ECS signaling and lower 2-AG. Polyphenols like curcumin and green tea, EGCG, are potent tools that regulate the enzymes responsible for 2-AG synthesis and degradation, helping to reset the system. Intermittent fasting reduces the overall substrate needed for 2-AG production, and plant adaptogens help the ECS recover its rhythm, reversing lipid overload. By seeing insulin resistance through the lens of the ECS, we open new doors for treatment and prevention. The new model confirms that insulin resistance is fundamentally a lipid signaling disorder. When 2-AG stays elevated, the ECS misfires, causing metabolic software error. When we restore ECS balance by rebalancing lipid inputs and controlling infection, the system recalibrates. Glucose sensitivity returns and metabolism stabilizes effectively. This presents a new frontier for precision nutrition therapeutics, focusing on the root signal, the endocannabinoid system, to re-engineer human metabolism.